inhibited, we can inhibit the whole disease mechanism. So this is the thing. So here we can classify it into structure-based design or ligand-based drug design. So the CAD is coming in picture. So these are the things we are making. And when COVID comes, it is like a long process to identify the drug. So initially they started using the FDA repurposing, and from now there are so many other strategies they are using. So before going to the COVID thing, we want to know what is bioinformatics. Bioinformatics is the like a field of science in which biology, computer science, and information technology merge to form a single discipline. So here we can omit biology, we cannot omit computer science, we cannot omit the information technology, because here we are studying the basics of biology, we are implementing the biological problems, we are overcoming it, we are using the technology with the computer science. What are the algorithms, what are the softwares, everything dealing with the computer science and information technology to gather the data, to store the data. If like only the gene sequence, from there how we can implement the sequence to structural bioinformatics, how it is enabling, because from the DNA they will isolate the sequence and they will deposit it in GenBank or any other database. From there we can collect what the bioinformatics will do. So here we want to check about somebody is checking and depositing the sequence in GenBank or something. How we will find out that sequence? We can take it as a model. So here is just like an example. We are always telling like humans are closely related to chimpanzees. So how they will identify? They are thinking like the sequence of the monkey, the chimpanzee they are taking, the sequence of the human they are taking. They both are like analyzing the sequence similarity, and they are checking that chimpanzee and humans are 97-96% similarity. So this is like a genetic evaluation, how it transforms from 1 to 1 over the years and it forms the human. So this is like a sequence level, how it is similarity. If you think about only monkey, it is around 96. If you think about even chicken, we have around 60% of similarity. So this takes like a multiple millions of years to transformation of genetic evolution. So this genetic evaluation, how we can determine if X or Y is submitting the sequence in the GenBank, we just copy the sequence and we are putting into the protein blast. From the protein blast, we are clicking the what are the available proteins, what is similar to that particular protein, and what is SARS-CoV-2. So nothing we don't know. So suddenly, biologists give the term COVID-19 and they given this is the sequence, and from there we don't know what is the protein structure, how it will be, what are the mechanism, everything we don't know. So in that category, we take in the sequences, we converted DNA to RNA, DNA to protein, or RNA to protein sequence, and from there the previous method, whatever I told, template matching, based on that template matching has done, and after that, based on the template matching, the homology models are developed. There are so many proteins available in the SARS-CoV-2, and each protein has modeled. Some experimental persons have crystallized or NMR structure. So they're like different techniques are implemented to find out the realistic structure, so that if we are getting the realistic structure, we can get the more reliable molecules that can bind into the particular proteins, so that the protein will get inhibited, the disease can be inhibited. So how the molecule can go and bind, how to check? So here protein DNA, the first picture, there is protein in red color and protein in blue color. So there are two proteins which can go and bind to each other. So it is the protein A and protein E is reacting when it is getting binding. Another is like a DNA ligand binding. So DNA drug. So this is like if you want to design a drug which can go and bind to the like DNA major groove binders or minor groove binders, how it will go and bind into the DNA itself. So the disease can be inhibited, or if the DNA can propose some problem, how it can get repaired. So these are all the DNA drug interactions. The final one is like a most common thing, is like a protein ligand interaction. So we will find out a drug which can go and bind to the active site of the drug molecule, and how the conformation is shifted from the initial to final binding. So this is like a protein ligand docking. So these are the main thing. And this is like a most successful technique like molecular docking, because we are like there, as I told, there's a billions of drugs are available in the market, and there are so many databases. So we don't know which molecule is going to bind with the protein. In that case, the molecular docking technique, which help you to say that this particular molecule is the apt molecule to go and bind in the active site of the protein. This can be anything like this ligand can go and bind with the DNA or protein or polymer, whatever. So this is like a more reliable, more close to experimental. So we can, instead of going for a, suppose if you want to test millions of compound in experimental, that cost huge millions. So this is like a molecular docking, which in like 5 days or 6 days, we can find out all the millions of molecules with the one single protein, and we can find out, oh, okay, this is the molecule which we are going to find, which we are going to test experimentally. Like that, we can find us. So this is like a molecular docking study. Another study is interesting. It's like a molecular dynamic stimulation. This molecular dynamic stimulation is the molecular docking is like a stagnant pose. We can check with a stagnant protein which cannot able to move, or it can move little bit for conformational shift. But when the protein model or when the protein ligand interaction happen, we have to check in the structure. In that box structure, we are filling the water models. So that water model will react with the protein structure and ligand molecule. It will show the realistic part of the dynamic studies. So the protein, when it's getting moved, the ligand molecule may come out, or it can go and bind in more tight mode. So in that case, we can find out whether the ligand molecule is apt inside the binding site, or it is ejecting from the binding site. If it is ejecting, the drug molecule is not good. If it is good, the binding interaction will be more stronger. So these type of realistic interaction we can find out using the molecular dynamics. It is based on the calculation with the time dependent, temperature dependent, and pressure dependent. So in this box, we is fixing that like up to 100 nanosecond how the protein is reacting. Up to, in the temperature, suppose if you are keeping about room temperature, we can set the temperature to 300 kilokelvin. And if you want like a human body temperature, we can set it to 310 kilo. So this is like a temperature setting. And if you want to give large, because the protein is should not go out, so we are keeping some pressure. And this is like a highly accurate technique to find out the protein movement inside the simulation box. So these are like a common techniques which I am going to speak in the further slides. So I am just giving the introduction here. Now coming to the main picture, like SARS-CoV-2. So this is like a, when we are getting the new term NCOV 2019, and the few renamed into SARS-CoV-2. So we are still like from that point to now, people are searching for the new drug molecule which can able to go and bind with the SARS-CoV-2 interaction, and it can kill the virus. And so what is SARS-CoV-2? If you think about what is SARS-CoV-2, it is like a large family of virus, and it is mainly infecting the humans. And sometimes we have the news like SARS-CoV-2 infected the lion, some like bats, camels, like that. So it is like initially it is a SARS-CoV is a single family, which is around 2000 or 2005. The previous virus came, but it is like a caused common cold to pneumonia it caused. But this particular SARS-CoV-2 is like more severe, and it is like it caused much death rate also. It is caused more infection. It caused like additional diseases also. So these are like more like updated version of the previous version, we can say. So if you see about the how it emerged, people are telling there are multiple stories behind the SARS-CoV-2 in like where it comes. First, they told Pangolian from where the SARS-CoV is initiated. Somebody told from bat it comes. Somebody told from camel it migrated. So these are the things. But we are getting similarity from everywhere. We are getting similarity from bat coves. We are getting from Pangolian virus. Some similarity we are getting from the MERS-CoV, like camel. So we are just in confusion dilemma whether where it come realistically, where it come. There are so many assumptions. Still now they are coming something like from the Pangolian it may arise more. So but we are getting more similarity. That is the confusion. Still there is no clear evidence what is the intermediate animal host from where it is initiated. So if you are thinking about COVID-19 timeline, so first in the mid of December, we have the news like we are having some virus is spreading, and there are some people are getting death. So we just have the news, and by the December 31st of 2009, who has announced that there is like the disease going to name as NCOV 2019 or COVID-19. And from the first COVID death has reported officially. And who reports first case outside wave, almost like Omicron, which didn't cause much things. But if you see about the picture, it will show how the spread is like it went out dark, how it slowly started from one place to one place, how the dark emergement from one location, like especially if you see about Delhi, Chennai, Bangalore, those areas like severely implemented, and it goes in dark. So when the beginning, when the people got how the common symptom or eventual symptoms, they got the fever, dry cough, bad breathing, and what it resulted, somebody got like pneumonia, kidney failures, gastro disorders. In addition, they got like somebody got like diabetic, somebody problem with the cancerous cells. And there are some people told like what you have to do and what you don't have to do. And then if you think about economic condition, whole like share markets around the world has dropped drastically. And if you think about the colleges, school, life science institute, like it is showing the data how much we have suffered in the beginning. And now we are thinking about COVID-19 drug development. So people around the world, all the researchers from small research institute to big research institution, funders, everybody have an eye to develop a drug which can go and inhibit the SARS-CoV-2 so that we can have the relief. So first, initially, they have find out, because if you find out a drug, we don't know whether it will work in a single shot. So people start with the there are like 1,800 approved drugs. So they have taken the 1,800 approved drugs. They tested all the drugs one by one, whether it will work with which target, whether the virus is getting infected, inhibited or not. Then they slowly check about what are the protein targets. If it is work, how it can like they can increase that activity of those molecules. And then there is still now we don't have any approved inhibitors. And how they can treat. So or suppose if you're having fever, doctor given some fever like based on the symptoms, they will give cure medicine for fever. If you're getting headache, the people doctor will give you the syrup for the headache. Likewise, based on the symptoms, they are giving the drugs. So if you think about SARS-CoV-2 life cycle and drug targets, so SARS-CoV-2 is having 4 structural proteins, 16 non-structural proteins, and 10 accessory proteins. If you think about structural proteins, it is basically it is working on the structure, like how the protein is like a virus is getting assembled, how the things are getting packed inside the virus are packed inside the cells, how it can eject their DNA or RNA and goes outside, and from there how from where the proteins are getting assembled and then again packed and get released. So this is the main process of structural proteins, and non-structural proteins is mainly placing the role in the mechanism level. So if you think about the life cycle, which I have given, the virus getting attached with the S protein like spike, it is getting into ACE2 protein, and once it got with the ACE2, it is going inside the cell, and from there viral RNA is getting released. From there, the human or ribosome is getting attached. From there, the polyprotein 1A and polyprotein 1B is proteolysis. Here, the main protease is getting inserted from the polyprotein, and from there, several fragments of protein is fragmented, like NSP7, 8, RDRP, like ribose transcriptase, helicase, NS9, methyltransferase. These are the proteins which are like doing their replication process, and from there, from it is going into the Golgi complex nucleus. From where the structural proteins are getting assembled, and finally the viral assembly is happened. The mature virions will exit the cell. So this is like a process of life cycle. It happens. So from here, we are thinking about how the dependency of each protein, because these many proteins are there. If one person want to work on all the proteins to find out a drug, it is very difficult. So the persons, like researchers, have identified the functionally important proteins to work on those proteins which can inhibit the protein. So here, this is the same Same thing with SARS-CoV-2 enters inside the cell and molecule. The first step. This is like an animation which shows the how the it goes inside the cell and how it is releasing. The second picture shows the RNA enters into the cell and change. It's like a polypeptide. This is like how the ribosome and viral proteins RNA is cleaved. And the final thing is like assembling the new proteins and how the new virus is coming outside the cell. And we are thinking about three proteins, right? One is like structural proteins, other one is like non-structural proteins, others is like accessory proteins. Actually, people are not, most of the people are not thinking. So the people who is already in disease is going to for the death level condition. So for that, these accessory proteins are main role. So we thought what is the reason behind that? How can we find out those? What is the reason why the people which are having the diabetes are going for death very easily? So when we find out these accessory molecules are like closed with the SARS-CoV-2 and MERS-CoV, and there is like if you see the OR of 3A, 4A is missing, 4B is like these are like assembled. This particle is particularly assembled to make it virulent, more virulent for the SARS-CoV-2. This evaluation is clear. It is from the ancestors of SARS-CoV and MERS-CoV. And this is also these accessory proteins find out. We are just checking the protein-protein interaction network mapping to find out which type of what are the proteins which is going and binding. So many network interactions are found for the each and every protein of the SARS-CoV-2. There's suppose if you're thinking about as previous COVID protein and it protein interactions which occurs between the normal human and cancer patient, we find out some protein which is like we are calling it as cancer drug targets. But these proteins, these virus proteins, are going and binding with the drug targets. So people will consume some drugs for their cancer survival. So these proteins going and directly binding to the cancer drug targets. So ultimately their cancer tendency is increasing because the suppressment of drugs is getting problematic. And here the protein-protein interaction that happens between the SARS-CoV-2 proteins and cancer proteins is suppressing the activity of the drugs, and it is the disease is containing both. So you can see the yellow round. So those are the cancer drug targets which is connected by the accessory proteins. So these are the results which shows that cancer proteins are directly interacting with the SARS-CoV-2, and that is the reason why the cancer patient, diabetic patients are getting severe after the post-COVID things. So this is not about single protein. Then we analyzed all the 10 accessory proteins. We find out several proteins which are having the direct interaction with the cancer. So here for the presentation, I have given some of the proteins which like cancer drug HMOX1, Mark2, Mark3. So these are the particular protein which are well known for the cancer drug targets, and especially the breast cancer, colon cancers. These are the things which is known for this particular drug target. So if you think about here we have shown only the picture live manner. We check about how the protein is getting interacted lively to with Mark 3. It is in the below level. So the strategy of SARS-CoV-2 is very interesting to particularly block the cancer proteins. So these are the interacting patterns. What are the amino acid role which each protein like OR of 3A, what is the amino acids interacting, and for the OR of 9B, what are the amino acids interacting, and OR of 9B, what are the amino acids interacting. Even the protein of OR of 9B is same with Mark 2 and Mark 3. There is huge similarity seen, but the interaction patterns is completely different. So these are the findings we have find out. And one more thing interesting I want to tell, there is a protein called OR of 9-3, like the pink color. This protein is going to bind with HMOX1. So this is like a heme binding protein. So this particular OR of 3A is exactly going and binding with the heme and intake the heme ion from the human protein. So it is like an energy barrier, how the heme ion is taken from where it's taken. So he take the heme source from this particular protein. So we will stop enough for the accessory proteins. There are so many other works also there. Let me continue with the non-structural proteins, because non-structural proteins are little more, because it is more involved in the mechanism line protease, NSP14, that is N7-N-tase. So these are the proteins. Why I have chosen these proteins to my research work? This is because these three proteins are having mechanism which is main role for the thing. Apart from this, there is one more protein called spike. That is like a structural protein, but it is having the main imperial role. But lots of other works are already have gone. So I have just omitted those spike protein, and I have gone with these three proteins. If you think about NSP3 domain, see, it is like a large protein. NSP3 protein is the largest protein after spike. So it is having main mechanism. It can go for the ADP ribosylation activities, which can introduce the induction of interferon. So once this particular thing happen, the substrate will go and bind to the host protein by the modification of the substrate which delivers. So due to this, interferon response will get dumped. So once this happen, it is like immunologically the human will get weak, and the mechanistically the virus will get more dominant. So here the inhibitor is the main role. If you develop an inhibitor which can go and bind with the viral macro domain, which can also replace the ADP ribosyl activity, that particular drug can be a better drug and can be also helpful for the human, because it is suppressing the immune genes. So this is like a particular protein, and so this is like a ADP ribose. Once it is going and binding, you can see there is like a L-shaped binding pockets available. So the ADP ribose it is like a natural natural cofactor which is in binding with the particular L-shaped packet. So there we need to design a drug which can go inside an L-shaped binding packet, which can replace like competitive inhibitors. We have to design a competitive inhibitor to go and bind inside the ADP repose, and it has to go into the which we don't want a different orientation, because this is like a L-shaped binding packet. We need a L-shaped ligand. The particular shape of the ligand should be there, so that we can get into the replacement, because it's like a competitive. So here, if you compete, this mechanism will not happen. The ADP ribosyl protein will not go for the unmodified ADP ribose. So this mechanism we have to stop. This is the main goal. So we have taken the sequence NSP3 MAC1, and we have think what is the ideology to develop the thing. So if SARS-CoV-2 is having 24 micromolar in like activity, SARS-CoV-2 is having 10 micro. So it is the level of activity is more. So to find out a drug, it is more imperial to go for this particular target. So if you check about the this is like initially how it is like told L-shaped. You can see the first picture MAC1 domain. There is a pink color rotator. How the L-shaped binding pocket is located. When we just plug out the L-shaped binding pocket, we can see the ligand is placing with the things. So we can classify it into what are the acceptor region, what are the donor region, what are the hydrophilic and hydrophobic region. So we have to find out a drug which exactly matches these regions. So we have used several docking strategies like virtual screening, structure-based screening approaches to find out a drug. Here there is a green color. If you're able to see the green color molecule, which is called the substrate, which is ADP ribose. But we have find out some more molecules which exactly mimicking the structure of the ADP ribose. And we can see all the structures are placing in the L-shaped. So we have find out some molecules which go and bind with the L-shaped binding pocket, and it is having the competitiveness than the ADP ribose. This orientation, we call it as conformational orientation. This orientation is exactly matching or mimicking the exact ADP ribose. This cause, if you give the drug, it can go inside and depth, and it can replace the ADP ribose, and by placing it position inside the active site. So the new drug can actively work on the MAC1 domain, NSP3 MAC1 domain. So we show the what are the interactions, what are the amino acids. So exactly we got the what are the, if you think about the A is the ASN 244, we have got the same interaction pattern for all the molecules from the gene database. We used some quantum mechanical study to ensure the activity, to ensure the interactions are realistic, because If you think about some molecular modeling, molecular mechanics force field, it may not that much highly accurate. So we used quantum polarized ligand docking, like quantum charges we have taken and import the quantum charges to the ligand to ensure the charges level. So because in quantum charges the accuracy is more, that's why we are used the quantum charges, and we have find out these molecules. So as I told, I have used quantum charges. We can think about like what is the electron capacity, because we are like A, B, C, D, E, F. There's like a six drugs. If you see about first four drugs, the pattern of the ligand is same, L-shaped, but the F is different. If you see, the head part of the A, B, C, E is same and the F is same, but the tail part is different. So it may not work on that particular thing. And if you think about HOMO, highest occupied molecular orbital, ligand is reacting inside the water models, how in the water model box. So in 100 nanosecond, these molecules are able to bind strongly, at least with two hydrogen bond is holding always. In average is holding at least two hydrogen bonds. If you see about the previous pictures, so there are so many hydrogen bonds, but at least two hydrogen bonds are following it up till 100 nanosecond of simulation. And the energy barriers are strong in every cases of all the ligands. And sometimes I can say the energy cases are better than the ADB reports. So in this ready, we are representing these molecules are better MAC1. You can see the polypeptide is go inside when the polypeptide is going inside, and once the polypeptide go inside, cleave the peptides, so that the new proteins will come out like the NSP7. There are so many other proteins. From here only it will start. So if you block this particular protein here itself, those proteins will not get erased. So this is the condition. We have to stop the viral replication, means we have to stop the N7 NSP5 main protease. We call it as main protease. So these protein has to get inhibited. So from China, we thought we can find out the solution also from that. At least like a 1 microsecond MD simulation. So this is like a big simulation we have tested for the previously reported N3 bound report, and also for the apoprotein. We have found out several things. If the protein is getting the apoprotein, there is a huge dynamic is seen. Due to this, conformational changes are more in the apoprotein. Apoprotein is only protein. When the inhibitor is going and binding, or inhibitor or peptide or anything is going and binding, the protein conformation changes is getting arrested. If you see the RMSD between the red and black, we can see there is a huge changes. The black is like a ligand bound complex. It is getting, the conformation is getting arrested. The RMSD count is getting decreased for the black, and for the RMSD counts is increasing the vibrancy up to 0.6 or 7. The frequency is going up and down. So this confirms that in the apo condition, the protein is having more functionally thing, and when the ligand goes and bind, so this can be a perfect drug target. And if you design a drug which can go and bind to the that polypeptide entrance, it can be a good drug for the viral replication inhibitors. So here we cannot use the any non-common strategy to develop a drug, because protein is very having so much of conformational changes which occurs in the event. So in the 1 microsecond conformations, we have taken each 50 nanosecond, we segregated the protein conformation different. And also we have taken the template protein. So 21 conformations we have got. Those 21 conformation we have got using the trajectory analysis. And from the 21 conformation, we have go for the heat map analysis. We have identified there are some major changes that occurs from the 21 conformation. So we have to find out a drug which can be active in these 21 conformations. Identifying a drug based on single conformation is getting easy, but we have to find out a drug which can go and work for the 21 conformations. So in these conditions, we are checking about what are the favorable conditions to work out. So we found out some molecules which can work on all 21 conformation. I will tell how it can be with 21 conformations. So these are the molecules which go and binds with the all the 21 conformation. There is some criteria required to how can identify the 21 conformation can be inhibited by these. So we set up some criteria the docking score of the should be able to show the binding energy of minus 35 or 40 below, we are omitting those compounds. By this criteria, we have scrutinized all the molecules, and we also need like if it is showing good interaction, if it is showing good scoring, we are not just checking. We have used one interaction profile for polypeptide cleaving cysteine 44, histidine 41, cysteine 145. These are the main amino acids that has to go for the interaction. If a molecule is going and binding with the cysteine 44, histidine 41, and cysteine 145, then the polypeptide mechanism will not occur. That is the reason we find out a molecule which go and bind readily with these amino acids. So we call it as like in main protease, the conformation, the traditional Chinese medicine molecules are very bigger molecules. So we thought the paper has reported if the molecule is able to cross S1 site and S2 site, then the molecule is good. So surprisingly, we got the molecules which acts like a S1 site and S2 site. In dissection things, we find out these molecules are realistically whatever we have given the criteria, everything it filled it, and additionally in the S1 site and S2 site. So those movements also we have allocated. So finally, if these molecules are introduced for the main protease, then it can be a realistic inhibitors for it can stop the polypeptide cleaving, and that carry forward the polypeptide cannot go inside the main protease, and several proteins cannot formed. So this replication can get blocked. And as I told previously, we have several criteria, I told, right? So this is like a zone of inhibition, whether the molecule is realistically able to inhibit or not. So in experimental, we are calling it as like zone of inhibition, like in microbiology, you people may know. So like that, we have find out that some concept how the in silico level, how the zone of inhibition can be represented. So we set the criteria for one side binding energy, one side docking score, how the zone of inhibition caused for each molecule. So we find out some molecule, as I told, it should be above 8.5 and it should be above 40 like that. So every molecules in each conformation satisfy this criteria and the zone of inhibition is formed. So these TCM compounds can be a better inhibitors for the main protease. And this is like a molecular dynamics for the ligand. We find out all the molecules are having the stagnant anything except the TCM 17404. It is having some ejection problem, maybe due to that ligand is getting out and coming inside. So we thought maybe this is not standardized molecule. So in realistic condition, we have omitted. In the publication also, we mentioned that this molecule don't much not having the tendency in the lively dynamic conditions. And these are the energy calculations pattern, like how the energy pattern can be inhibited, like van der Waals energy, binding free energy, electrostatic potential energy. So these are different types of energy, how the particular molecules are showing the energy levels in the live dynamic condition. So these are showing the like because this is like a dry lab and computational, we have to be more depend on energy levels to find out the molecules whether working with that or not. So in this condition, we are checking about how the binding free energy calculations are able to show the energy levels, which can, if the energy levels are more lower, then it is more good. So if we find out some molecule which is having more lower energy level, lower means if it is minus, we are telling as lower. So we can see all the energy levels are in lower level. So we find out these molecules are better in terms of docking, dynamics, conformational changes, amino acid pattern, interaction pattern, active site analysis, everything. So we are declaring these molecules can be a strong pro inhibitors for the experimental things. So next thing is like another interesting thing. There is a cap. It is showing, right. So there is a virus is also having a cap like strategy. So this N7 MTase is having a capping system. So once the RNA is getting inside the host cell, it will the human immune system is like a very clever thing. So to tackle the human immune system, there should be a more clever thing which virus is following. There you can see like a cone-shaped thing. So the viral RNA goes and check whether it is human or foreign particle. This cap system will say, no, I am a human particle. I am a human thing. So don't disturb like that. It is saying honestly, and that is the reason in initial condition it is not able to show the inhibition in initial point itself. So if you inhibit this particular protein, the human immune system will be clever in the initial stage itself. So that is the reason we have chosen this particular protein NSP14. We call it as N7-methyltransferase, that is MTASE. This capping system has to be properly checked for the inhibitors. Then only a beginning stage of the virus infection itself we can inhibit it. So these are the mechanism what it costs. So this is, we call it as viral capping mechanism, viral RNA capping mechanism. And this clever mechanism is not not available in the SARS-CoV, but it is available in the SARS-CoV-2. And for this protein, the protein is not, till now, their protein is not solved experimentally. So we have chosen the protein, this around 526 amino acid. And if you think about 576 amino acid, we call it as NSP14. And from 1 to 287, it is called the exon. And from 288 to 526, that is called the N7-MTase. So this particular protein, we need only N7-MTase. But without the exon, the protein N7-MTase is not getting active conformation. So we need both the conformation, whether with we need exon also, we need N7-MTase also. So we get both domains we got. So this is the thing we need to check on protein modeling by saying whatever I told in homology modeling thing. The similar way from the SARS-CoV, we have developed the protein exon, like whatever I round, it is like N7-MTase, and it is like a exon is different. But our binding pocket is in N7-MTase, and exon is not a drug target. Only N7-MTase is a drug target. So in this condition, we have checked about what are the similar thing. We have find out similar thing to check about the thing. We found out one loop also extra which is found. So we thought that is the capping system which is not available in the SARS-CoV. And there is like a cofactor conversion is there from G3A to G3T. So this cofactor is the main role which is raising in the particular protein more functionally. So if you are blocking this cofactor, then the protein mechanism will get blocked. And also we can find out some inhibitor which can go and bind with the particular cofactor thing. So I have an intention to block that cofactor. So we checked what is the cofactor interactions with the N7-MTase. And we found out by structure-based method, this is like a structure-based strategy we have followed with the same TCM database. We found out here it's like a not more confirmation changes are not seen. So we take in the stagnant pose and we checked the structure-based screening, which gives some 6 molecules which can go and bind with the same G3P interaction pattern. And we find out these molecules can very much strongly bind with the GTP site. And that's the reason we have chosen these molecules to be a eminent N7-MTase. We have checked with the molecular dynamics also how the particular protein is getting, whether with a cofactor, how it is simulating with the molecules, if it is anywhere some shift is checked up. We have found out none of the molecule is not able to eject. So we found that these molecules are having the strong interaction pattern and in a dynamic condition, unstable condition. Then once we confirm this, we want to find out some system level analysis to check whether it is like denaturing the protein content or like that. So we find out only protein molecule F is not able to contain the denatured conformation. So apart from that, every protein, every ligand molecule is able to find out the strong adjustness. So once I am closing all the work, so these are the research publications from COVID. So these all my research works get published in different journals. And I have recently edited 2 books on COVID-19. So these are the 2 books. And thank you very much. If you have any doubt, now you can clear. And if you have any other doubt, you are welcome to write an email to this email ID. Thank you.
Thank you, sir, for your valuable words.
Thank you.
Dear participants, if you Hello, sir.
Yeah, welcome.
Sir, how to find protein-protein interaction in multiple sequence alignment?
Multiple sequence alignment is different and protein-protein interaction is different. Multiple sequence alignment is to check the given protein of sequence, what is the similar protein from the available database. But in terms of protein-protein interaction, we have to first initially model protein and we have to find out if the protein is already available in the PDB database or AlphaFold or any other database. Then we have to go for some servers like Rosetta, Robeta, PatchDoc, HackDoc. So these are different softwares already available. So those softwares we have to take and we have to find out one protein like chain A and chain B because we have to differentiate the software that this is like a protein A and this is like a protein B. Then we have to start the strategy like we have to clean the protein. We have to find either the protein is having the steric classes or not. We have to prepare it properly and we have to find out the protein-protein interaction between those proteins. So these are like entirely different things. Are you able to understand?
Okay, sir. Good evening, sir.
Good evening.
Myself Hashrat. I want to do MS in foreign, sir.
Okay.
Full scholarship. Is there any procedure or any competitive exams you want to study?
Oh, actually, this is like okay. There is which country you have to choose. This is the main thing. Suppose if you are thinking about well-grown countries, you have to clear the IELTS or TOEFL. Then you have to find out some institutes. You have to check about which area you are more fond of. Then you have to apply that professor. That dear professor, I am interested to PhD with you or MS with you. So is there some option to give some funding? So they will suggest you the better way or else you can go to some other university. You can find out whether the scholarship they are providing. This is the other way. Otherwise there are some government funding fellowships, the country based fellowship. Indian government is not giving for abroad, but other countries they are like for Korean they are giving like brain pool fellowship, so they are giving for the MS or a PhD. So those things we can check and we can explore the things. It's depend on country
Okay,
wise.
thank you. Sir, which country is better, sir, for pharmaceutical technology?
Oh, most of the people are going for the US and Europe, but more advanced things are available in US and Europe only. And still nowadays China is also rocking in pharmaceutical technology. If you are able to go China, US, Europe, it is welcome to go.
Thank you, sir.
Anybody can ask any doubts. I am happy to clarify.
Hello, sir.
Yeah.
Sir, good afternoon, sir. Myself,
Good afternoon.
final year pharmaceutical technology, sir. Sir, hello, sir.
Yeah.
Sir, if you're telling MD stimulation take times for 100 nanoseconds, I have a MacBook Pro MD processor, sir.
Okay.
If I run MD stimulation and how much time it takes, sir? Can I do it from a laptop?
Actually, MD simulation is like a highly configured system you have required. MD simulation and quantum mechanics, these two are having problem with the systems because it is not about like 16 GB RAM or 32 GB RAM or like that. We need at least like 500 GBs of RAM. So highly configured systems we required to run more than 100 nanosecond or 1 microsecond. If you run just for 10 nanosecond, if you're having some 16 GB RAM system, better go. If you load the system in MacBook, sometimes it will get hanged and it is because it is like a long time. You cannot switch off the system for at least 10 days, next 10 days. So it is not advisable.
Thank you, sir.
Hello, sir.
Yeah.
Myself, Sneha, final year Pharmaceutical Technology, sir.
Hello.
Sir, your image presentation slide, easy to understand and look great, sir.
Thank you.
Sir, how to DPI image, image like that? Tell us
Sorry,
something about that, sir. How to create DPI image like that? Tell us something about that, sir.
You mean animations or normal picture? Snega?
Sir, how to create DPI image like that? Tell us something about that, sir.
Okay. So this is like most of the things are we are creating with the softwares, and we need like a strong graphic card based system because most of the thing we are depending on scoring and pictures. So picture has to be more clarity to represent our pictures. In that case, we need a graphics cards to display the more clarity pictures. Once we are taking the pictures, we are just making it more with at least 900 dpi to make it more. If you enlarge the picture, the pixel cannot be reduced. So this is the main thing we are doing. Apart from that, we cannot edit the picture because journals will not accept the edited pictures. So that is the thing. We are not doing anything special. It's just graphics card is doing everything. I hope I have cleared your answer. Are you okay?
Thank you, sir.
Okay.
Sir, what is DFT calculation?
Density functional theory is like suppose we are applying the quantum mechanics. So in quantum mechanics, there are different strategies are there. So by using the single atom charges, like if it is a drug, it may have around 150 atoms. So each atom has to check for the realistic charges because some places we may have the pseudo charges. The pseudo charges means some places it will be neutral. So if the reaction has to occur, the atom has either positive charge or negative charge. Then only the interaction is possible. To avoid the pseudo charges, we are applying the DFT calculations to understand from where the electron transfer is occurs. Like I displayed the one picture called HOMO LUMO, like where is the highest occupied orbital. From there, the electron is getting transferred, and from LUMO, how the electron transferred is getting accepted. So these are the different things which can be tested using the DFT, density functional theory calculations.
Thank you, sir. Dear participants, if you have any queries, just post it in our chat box.
Sir, myself
Uh,
P. Vaishnavi from Pharmaceutical third year, sir. I had
Hello.
one doubt, sir. Hello, sir.
Yeah. Yeah. Tell me. As I told, it is like a drug target. So we have to find out that. Suppose I told right, based on similarity, we can choose the protein, which is like in Ancestr's protein, how it is. In the condition, suppose NSP3 I am taking, it is already having in the SARS-CoV. So we know that that particular protein is for the NSP3. By that we know the particular function of NSP3 is for immunovirulent. So if you take about that particular protein, we can go for the that particular protein for the dreadful thing. By this we are choosing. If you want to exactly know, we have to go for the about the NGS next genome gene sequence. By that we can go for the gene level, find out those genes we can extract, and we can convert those particular genes into proteins, and then we can carry out. So these are two methods are there. If you are not able to ask.
Sir, what is
Yeah.
RMSD and what are the interpretation of RMSD?
So RMSD is like expansion is like a root square mean square deviation. So we are checking about the dynamic simulation, right? So dynamic means then we are noted. Based on that, we are developing a graph how the movement of the protein is getting excited state, or again it is coming to the ground state, or not. Like that we are checking. That is called RMSD.
Thank you, sir.
Okay. Is it some Mukesh? Is it possible to identify the interaction of amyloid with unknown protein in silico like MED simulation? Yes, amyloids can be checked. Already they have several works, amyloid interactions. But initially we have to go with the molecular docking strategy. And based on the docking pose, we have to find out the interaction with the MD simulation. Even some membrane, membrane-based simulation also has been tested with amyloid proteins. Sure, for writing review article, I am happy to help. Whenever you need help, just ping me with the email. How is the Vishwa? How is the drug design used in pharmaceutical technology? It's not, drug design is a big field. And pharmaceutical technology, actually, you people are only thinking using the drug design more than me. So you will use more for you. Maybe you are in beginner stage. When we are going in more advanced stage, you will use more. So what is that? Ajit Kumar final year. How to find out the peptide that has antimicrobial activity? So for checking, we can in dry lab we can predict the activity, but not. So if you have to really find out the peptide as an antimicrobial activity, you have to synthesis the peptide. You have to go for which organism. Then you have to go for MIC activity or anti-biofilm. Those things you have to check about the experimental only. So Sweta, is there any method to find out which type of molecule get amino acid, which type of so actually I told there is one picture I to have the that particular aromatic ring, the aromatic amino acid, and they will see this is hydrophobic. And if there is like a hydrogen, it will go and bind with the oxygen atom only. If it is nitrogen atom, it will go and bind with the oxygen atom. If it is carbon atom, it will go to bind with the oxygen atom. Like acceptor means where it can donate the atom, accept the atom, and when it is donate, it will accept like that. Then is there Janani? Is there any principle which is common in pharma? Sorry for this question, I don't have an answer because I am food technology. I am not aware, but people are using some of the food molecules for the pharmaceuticals. Nowadays, food as medicine also one concept is arised.
Thank you, sir.
Thank you.
Any more questions? Now I call upon Aarti to deliver the vote of thanks.
No duty is more urgent than that of retaining times. Good evening, one and all gathered here. It's given me an immense pleasure to deliver the vote of thanks for this wonderful virtual seminar. First and foremost, I would like to express my heartfelt thanks to our guest speaker of the day, Dr. Chandrabose Selvaraj sir, who despite his busy schedule has found time to grace this session. I would like to express our gratitude to Chairman sir, Correspondent ma'am, Admin sir, Dean ma'am, Principal sir. I would like to thank our beloved Dr. R. Praveen Kumar sir, Head of the Pharmaceutical Technology, and Professor R. Balasubramani sir, Head of the Department Food Technology for their guidance. I would like to thank our faculty members and students coordinator of this seminar for making this session a great grand success. Last but not least, I thank all the participants who accept our invitations and gathered here. Thank you all. Research is to see what everybody else has been and to think what nobody else has thought. Once again, I thank you for all your coordination today. Thank you.
Thank you.
Thank you, participants, for your coordination. Dear participants, kindly fill the feedback form. Then only certificates will be provided within a week. Feedback form is posted in a chat box.
Okay, thank you. Thank